Students, in this presentation, I'm going to tell you about one of the most common sexually transmitted diseases, that is syphilis. The causative agent of this disease, syphilis, is Treponema pallidum. Please see the spelling correctly and write correctly, because many times the students make mistake, especially in the spelling of pallidum. So it is Treponema pallidum, not palladium or palladium, it is pallidum. And this organism, Treponema pallidum, is basically a spirochete. See, in the grouping of bacteria, depending on their shapes, we have five basic groups, right? Bacilli or rods, cocci, and then Vibrio, spirillum, and spirochete. So, when we divide the bacteria based on their shape, we divide them into five groups, and this Treponema pallidum comes under the spirochete group of bacteria. Coming to the morphological characteristics of this organism, the Treponema is made up of a thin, slender body, a very long rod which is thin and slender, and this rod is not straight, it is a spiral rod. As you can see here, it is a spiral shaped rod, and the ends of this rod are tapering. That is, it is not the exact size as the center. It is tapering, it is narrowing down. So thin, slender, this is a thin, slender spirochete with tapering ends, and as I said, the body is not straight, it is made up of spirals, and usually around 10 spirals will be there in one organism, in one cell. And if you see the size, each spiral is about 1 micrometer in size, in length. So if there are 10 spirals, so the total length will be around 10 micrometer. So that is what is written here. It is 10 micrometer in length, whereas only the length is large, the breadth or the width is very, very thin. Only 0.2 micrometer width will be there. So they are thin rods. The length is longer, of course, but the width is very, very thin. Coming to their motility, they are a very actively motile organisms, but they don't move with the help of external flagella like other bacteria. They move with the help of a flagella which is running throughout the body of this organism. Running throughout the body in the sense, this flagella is present in between the outer membrane and the cell membrane of this organism. It is present in the periplasmic space of the body of this organism. So that is why the flagella is called endoflagella. It is not present protruding outside like other bacteria. It is present embedded inside the bacterial cell itself in its cell wall. And this is called axial filament or endoflagella. And as I said, they are actively motile organisms. In fact, the motility will be like a rotatory motility. The whole body will rotate round the long axis, and that is how they move. Just like a worm which you would have seen moving, the whole body contracts and expands while moving. So that is how spirochete also moves, using its whole body and rotating it with the help of endoflagella. And this organism does not form spores and it is not capsulated. Now, when we see the cultural characters of this organism, it is a little different from other bacteria again. Pathogenic treponemes cannot be grown in artificial culture media. You cannot use nutrient agar or blood agar or chocolate agar or LJ media. No such artificial culture media can be used to grow the pathogenic treponemes. Very rarely, sometimes they maintain this organism for a few days in a very complex culture media under anaerobic condition, not a very defined media again. using some reducing agent under anaerobic condition, you can maintain them for a few days, not for even months. But usually they maintain certain treponeme cultures in rabbit testis. In rabbit testis, they can maintain the culture, that too by serially passaging it from one rabbit testis to another rabbit testis. You can maintain it for quite some period of time, and this is how they have been maintaining this particular strain. Nicole strain is one strain of Treponema pallidum which was isolated from a patient in 1913. This is a pathogenic treponeme, and this has been maintained in rabbit testis by serially passaging it. And why am I talking about this particular strain? The reason is this strain is most commonly used for both diagnostic purposes and also research purposes in the laboratory. Of course, we have to conduct certain research in this organism to know its characters, to develop kits for detecting it in a patient, to take antigen from this organism for using it in the diagnostic kits. So all these are necessarily to be done. So which organism is taken for this? It is the Nichols strain of treponeme has been taken for this to carry out all these activities. Okay. Now, there are certain strains which are non-pathogenic nature, and they have been maintained for a short period in cell cultures. See, tissue cultures or cell cultures sometimes can be used. Cell cultures like Eagle and McCoy cell lines can be used along with fetal bovine serum and also some reducing agent. So you need to add a lot of complex components here to maintain this organism in the laboratory. They are not easily cultivable. They cannot be grown in artificial culture media. They can be maintained in rabbit testis. So all these constraints are there when we talk about growing treponema in the laboratory. Coming to the antigenic characters, though three different antigens have been identified from this organism, they could not study these antigens very clearly, except for this one. They could not study the antigenic characters very clearly because, as I said, they cannot be grown very easily. But why do they say that these three antigens are present? Because when these antigens are injected in human beings, we could see the production of three different types of antibodies. So from that antibody structure, they have identified that there are at least three different antigens that could be present in this organism. What are these antigens? The first one, you should remember it, is cardiolipin antigen. This is basically a lipid antigen. So this antigen is prominently present in this organism. So as I said, it is a lipid antigen, but instead of calling it as an antigen, I am going to call it as lipid hapten. See, you will study about this hapten and antigen in detail in immunology again. So let me not go into details about it. It is an antigen, but it is not a complete antigen. It is an incomplete antigen, and that is why it is called a lipid hapten. And when this antigen is injected in the human beings, it gives rise to a specific type of antibodies called reagin antibodies. Okay. Now, though this antigen elicits an immune response, though this antigen gives rise to the production of antibodies in human beings, still this antigen is not very specific in nature. Specificity andre enu, antigen jothe ಒಂದು particular type of antibodies ಮಾತ್ರ react ಆದರೆ ಅದನ್ನು ನಾವು specificity ಅಂತ ಹೇಳ್ತೀವಿ. But this antigen is not very specific. That means ಈ ಏನ್ antibodies ಬಂದಿದೆಯಲ್ಲ, this antibodies can react with some other antigen also. Apart from this antigen taken from treponema, these antibodies can react with some other antigen also. So ಯಾವ antigen ಜೊತೆಗೆ react ಮಾಡುತ್ತೆ? It can, this reagin antibodies can react with another antigen taken from beef heart. ಬೀಫ್ ಹಾರ್ಟ್ ಅಲ್ಲಿರುವ ಒಂದು ಆಂಟಿಜನ್, ಅದು ಕೂಡ ಕಾರ್ಡಿಯೋಲಿಪಿನ್ ಆಂಟಿಜನ್. ಕಾರ್ಡಿಯೋಲಿಪಿನ್ ಅಂತ ಹೆಸರು ಬಂದಿರುವುದೇ ಇನ್ಫ್ಯಾಕ್ಟ್ ಅದರಿಂದಾನೆ. ಕಾರ್ಡಿಯೋಲಿಪಿನ್ ಅಂದ್ರೆ ಲಿಪಿನ್ ಸ್ಟ್ಯಾಂಡ್ಸ್ ಫಾರ್ ಲಿಪಿಡ್ ನೇಚರ್ ಆಫ್ ದಿಸ್ ಆಂಟಿಜನ್. Cardio stands for, basically this antigen has been taken from beef heart. So E antigen, yen beef heart in the E antigen tegdidaro, this antigen is similar to this antigen taken from Treponema pallidum. So that is why they say this antigen is not very specific in nature. Okay, so you remember this particular antigen. Why? Because this is the antigen which we commonly use in the diagnostic kits. ನಿಮಗೆ ಲ್ಯಾಬೊರೇಟರಿಯಲ್ಲಿ ಡಯಾಗ್ನೋಸ್ಟಿಕ್ ಕಿಟ್ಸ್ ಇರುತ್ತೆಲ್ಲ, ಅದರಲ್ಲಿ ಒಂದು ಆಂಟಿಜನ್ ಇರುತ್ತೆ ಅಥವಾ ಆಂಟಿಬಾಡೀಸ್ ಇರುತ್ತೆ. ಈ ಸಿಫಿಲಿಸ್ ಡಿಟೆಕ್ಷನ್ ಅಲ್ಲಿ ಆಂಟಿಜನ್ ಇರುತ್ತೆ ಕಿಟ್ ಅಲ್ಲಿ. ಯಾವ ಆಂಟಿಜನ್ ಅಂತ ನೋಡಿದ್ರೆ ಇಟ್ ಇಸ್ ದಿಸ್ ಕಾರ್ಡಿಯೋಲಿಪಿಡ್ ಆಂಟಿಜನ್ ವಿಚ್ ಇಸ್ ಪ್ರೆಸೆಂಟ್ ಇನ್ ದಿ ಕಿಟ್. The next antigen is the group specific antigen. That means this antigen is present only in Treponema. Bere yava organism allu illa, Treponema genus alli ee antigen ide. That is why it is called group specific antigen. Whereas there is another antigen also which has been identified, that is species specific antigen. Treponema pallidum species alli matra iruva antigen kuda ide, that is species specific antigen. Basically this antigen is polysaccharide in nature. I said cardiolipin antigen is lipid in nature, whereas species specific antigen enideyo that is polysaccharide in nature. So these three antigens have been identified in Treponema pallidum. Coming to the resistance character of this organism, Treponema is a very delicate organism where it will get readily inactivated by drying or even raising the temperature to just 41 to 42 degrees Celsius in 1 hour. That means one hour olagade it will die if we increase the temperature where it is present to 41 to 42 degrees. Ishte raise madidare saku, it will die. See, it is this character which we sometimes exploit to kill this organism in the human body. Sometimes another syphilis infection ide anta adre ಏನು ಮಾಡ್ತಾರೆ, intentionally body temperature na increase ಮಾಡ್ತಾರೆ. 37 ಇದೆ ಎಲ್ಲ ನಾರ್ಮಲ್ ಟೆಂಪರೇಚರ್, ಅದನ್ನ intentionally 41 ಗೆ ತಗೊಂಡು ಹೋಗ್ತಾರೆ. So that if you maintain the body temperature at 40 to 41 for even 1 hour, the organism there is a fair chance that the organism will die. So ಇದನ್ನ ನಾವು fever therapy ಅಂತಾನು ಕೆಲವೊಮ್ಮೆ ಹೇಳ್ತಾರೆ. This was tried once. And even low temperature it cannot withstand. It will get killed at 0 to 4 degrees Celsius in 1 to 3 days. And most of the chemicals can easily kill this organism, like soap, arsenic compounds, mercury compounds, bismuth compounds, and even normal antiseptics what we use in clinics and even households. Idu elladakku idu easy agi saayutte. So fomites alli andre inanimate objects alli ee organism survive agalla. It will get killed very quickly. But you can maintain this organism for over a long period of time at a frozen temperature. Frozen temperature, that to 10% glycerol alli ಹಾಕಿಡಬೇಕು ಅಥವಾ liquid nitrogen alli mix ಮಾಡಿ ಇಡಬೇಕು. So ಈ ತರ frozen temperature alli ಇಟ್ರೆ you can keep it for even 10 to 15 years. Coming to the pathogenesis, the mode of transmission is usually by sexual contact, especially when it is transmitted through sexual contact. We call it as venereal syphilis. But it is not only through sexual route it enters. Sometimes when blood transfusion is carried out, if the blood is contaminated with syphilis organism, then through that route, that is through blood transfusion also, it can enter inside the human body. Sometimes from the mother to the fetus through the placenta, it can enter the fetus body. So transplacental route is there, blood transfusion, blood route is also there, and sexual contact can also transmit the infection from one person to another person. And usually it enters the body through minute cuts or abrasions on the skin or mucosa. Healthy skin athava mucosa nalli usually it cannot cause the disease. Mucosal layer alli enadru damages idre, cuts iddu, abrasions or cracks enadru idre, through that route it will enter inside the human body. And even when it enters inside the human body, it is not going to spread very quickly. Usually the site of multiplication will be the same as the site of entry. Elli entry ಆಯ್ತೋ ಅಲ್ಲೇ ಇದ್ದು infection first ಅಲ್ಲೇ ಕಾಸ್ ಮಾಡುತ್ತೆ. It's only if you don't treat the patient on time, then it can get spread to other parts. Otherwise, usual primary infection is usually at the site of entry. And the incubation period for this organism to show first sign of symptoms is about a month. 1 month ವರೆಗೆ ನಿಮಗೆ ಇನ್ಫೆಕ್ಷನ್ ಇರುವುದೇ ಗೊತ್ತಾಗಲ್ಲ. This is another negative point here, because even an infected patient cannot be detected for a whole month. So our time nalli he would be spreading the organism to other person. So what are the symptoms that you will see in a patient who has been infected with treponema? The symptoms can be divided into 3 stages depending on the severity. In the early phase or in the first stage, what you will be seeing is only a lesion. ಒಂದು ಗಾಯ ತರ ಇರುತ್ತೆ. Wherever it has entered and wherever it has multiplied, ಆ ಏರಿಯಾನಲ್ಲಿ you will just see a lesion, ಗಾಯ. And that lesion is called chancre. And this lesion is usually seen in genital region. As I said, the primary mode of entry is through sexual contact. So sexual contact alli entry ಆಯಿತು ಅಂತಂದ್ರೆ genital region ಅಲ್ಲೇ ಇದು infection ಆಗುತ್ತೆ. So ಈ genital region ಅಲ್ಲಿ ನಿಮಗೆ ಹೇಗೆ ಕಾಣುತ್ತೆ ಈ lesion ಅಂತಂದ್ರೆ it is called chancre. Now how can we define chancre? Chancre is basically a painless, relatively avascular, circumscribed, indurated, superficially ulcerated lesion. Do not miss any point here. It's quite easy. Painless ಅಂದ್ರೆ ಇದೇನ್ ನೋಡ್ತಿರಲ್ಲ ಇದೇ chancre lesion. ಇದು orally ಬಂದಿದೆ, ಇದೇ ತರ genital region ಅಲ್ಲೂ ಬಂದಿರುತ್ತೆ. So what you see here is the chancre. So it is painless. ನೀವು ಮುಟ್ಟಿದ್ರೆ ನಿಮ್ಗೇನು ನೋವಾಗಲ್ಲ ಅಲ್ಲಿ. Second is relatively avascular. That means you will not see any blood circulation in that region. Circumscribed. That means it will not be spreading. ಒಂದು ಏರಿಯಾನಲ್ಲಿ ಡಿಮಾರ್ಕೇಟೆಡ್ ಏರಿಯಾನಲ್ಲಿ ಮಾತ್ರ ಇರುತ್ತೆ. Indurated. That means when you touch it, it will not be soft. ಸ್ವಲ್ಪ ರಫ್ ಆಗಿರುತ್ತೆ ಆ ಏರಿಯಾ. Indurated. And superficially ulcerated lesion. Superficially ulcerated ಅಂತಂದ್ರೆ ಒಳಗಿನ ಟಿಶ್ಯೂ ಏನು ಅಫೆಕ್ಟ್ ಆಗಿರಲ್ಲ, ಓನ್ಲಿ ಔಟರ್ ಏರಿಯಾನಲ್ಲಿ ಮಾತ್ರ ಅಲ್ಸರ್ ತರ ಸ್ವಲ್ಪ ಡ್ಯಾಮೇಜ್ ಆಗಿರುತ್ತೆ ಆ ಏರಿಯಾ. ಸೊ ದಿಸ್ ಇಸ್ ವಾಟ್ ವಿ ಕಾಲ್ chancre. So this chancre in most of the patients, it will just heal on its own within 10 to 40 days. Even without treatment, just a scar will be left. Otherwise, the patient will completely recover within 40 days, sometimes even without treatment. And this lesion is usually a localized lesion, or it will be basically a localized infection. In 1/3 of the patients, the disease will not proceed further. Either the patient will heal on his own, or he will be treated on time, and then he will be healed. But sometimes after the primary lesion heals, still the syphilis organism would have survived. So in this case, what will happen? Slowly this organism will start getting spreaded through the blood throughout the body. So almost after 2 to 6 months, you will start seeing symptoms throughout the body. And this is when we call it as secondary syphilis. In primary syphilis, as I said, the infection will be only a localized infection. The lesion will be seen only in a particular region wherever it has entered. Whereas in the secondary syphilis, if the organism gets spreaded through the blood and reaches many parts of the body, then you will start seeing the symptoms of secondary syphilis. What is the main symptom of secondary syphilis? As you can see here in the picture, you will start seeing roseolar or papular skin rashes. Roseolar ಅಂತಂದ್ರೆ ಇಲ್ಲಿ ನೋಡ್ತಿರಲ್ಲ pink color spots ಇದೆ. This is what we call roseolar rashes. ಅಂಡ್ ಪ್ಯಾಪ್ಯುಲರ್ ಅಂತಂದ್ರೆ ಬಾವು ಬಂದಂಗೆ ಸ್ವಲ್ಪ ರೇಸ್ಡ್ ಆಗಿರುತ್ತೆ, ಜಸ್ಟ್ ನಾರ್ಮಲ್ ಆಗಿರಲ್ಲ ಸ್ಕಿನ್, ಸ್ವಲ್ಪ ರೇಸ್ ಆಗಿರುತ್ತೆ. ಸೊ ದಟ್ ಇಸ್ ಕಾಲ್ಡ್ ಪ್ಯಾಪ್ಯುಲರ್. ಸೊ ದಿಸ್ ಇಸ್ ಎಸ್ಪೆಷಲಿ ಸೀನ್ ಇನ್ ದ ಟ್ರಂಕ್ ರೀಜಿಯನ್, ಇನ್ ಎಕ್ಸ್ಟ್ರಿಮಿಟೀಸ್ ಅಂದ್ರೆ ಪಾಮ್ಸ್ ಅಲ್ಲಿ, ಸೋಲ್ಸ್ ಅಲ್ಲಿ, ಇಲ್ಲಿ ಪಾಮ್ ಅಲ್ಲಿ ನೋಡ್ತಿರಲ್ಲ ಅದೇ ತರ ಕಾಲ್ ಕೆಳಗಡೆ ಸೋಲ್ ಅಲ್ಲಿ ಇದೇ ತರ ಇರುತ್ತೆ, ಬೆನ್ನಲ್ಲಿ ಟ್ರಂಕ್ ಬೆನ್ನಲ್ಲಿ ಇರುತ್ತೆ, ಮುಂದೆ ಹಿಂದೆ ಎಲ್ಲ. ಅಂಡ್ ಆಲ್ಸೋ ಇನ್ ದಿ ಓರೋಫೆರಿಂಜಿಯಲ್ ರೀಜನ್ ಅಂದ್ರೆ ಬಾಯಿ ಒಳಗೆ ನೀವು ಆ ಟಾನ್ಸಿಲ್ಸ್ ಏರಿಯಾ ಇದೆಯಲ್ಲ ಅಲ್ಲಿ ಕೂಡ ಯು ವಿಲ್ ಫೈಂಡ್ ದೀಸ್ ರೋಸಿಯೋಲಾರ್ ಆರ್ ಪ್ಯಾಪ್ಯುಲಾರ್ ಸ್ಕಿನ್ ರ್ಯಾಷಸ್. ಸೊ ಇಷ್ಟೆಲ್ಲ ಬಂದಾದ್ಮೇಲೆ ಅಗೇನ್ ವಿ ಆರ್ ನಾಟ್ ಗೋಯಿಂಗ್ ಟು ಕೀಪ್ ಕ್ವೈಟ್, ವಿ ವಿಲ್ continue, we will start the treatment. So again because of the treatment the disease will get cured. So in some patients it will end with secondary syphilis. If you don't give proper treatment in the secondary syphilis stage, almost 40% of the syphilitic patients may proceed to the tertiary stage. And that is when we are going to call it as tertiary syphilis. So here mainly the cardiovascular system and the central nervous system will be affected. So you will start seeing andre heart alli and brain alli nimage effects kaanakke shuru agutte. So cardiovascular lesions will be aneurysms, chronic granulomata, meningovascular effects. Aneurysm alli the arteries will get affected, and chronic granulomata means where inflammation will happen, resulting in the accumulation of different types of immune cells. So ಒಳಗೇನೆ inflammation ಆಗಕ್ಕೆ ಶುರು ಆಗುತ್ತೆ, which will affect the meningeus region. Same way you will also see neurological effects like paralysis, mental retardation, blindness and shuffle walk. ಅಂದ್ರೆ ಯಾವಾಗ ಬ್ರೈನ್ ಅಫೆಕ್ಟ್ ಆಗುತ್ತೋ ಬ್ರೈನ್ ರಿಲೇಟೆಡ್ ಆಕ್ಟಿವಿಟೀಸ್ ಎಲ್ಲ ಅಫೆಕ್ಟ್ ಆಗಕ್ಕೆ ಶುರು ಆಗುತ್ತೆ. ಬ್ರೈನ್ ರಿಲೇಟೆಡ್ ಆಕ್ಟಿವಿಟೀಸ್ ಅಂತಂದ್ರೆ ಒಂದು ಒನ್ ಸೈಡ್ ಆಫ್ ದ ಬಾಡಿ ವಿಲ್ ಬಿಕಮ್ ಫಂಕ್ಷನ್ಲೆಸ್, ಪ್ಯಾರಾಲಿಸಿಸ್ ಅದಕ್ಕೆ ಎಲ್ಲ ಹೇಳೋದು. ಅಂಡ್ ಮೆಂಟಲ್ ರಿಟಾರ್ಡೇಷನ್ ಅಂದ್ರೆ ಮೆಂಟಲ್ ಫಂಕ್ಷನ್ಸ್ ಪ್ರಾಪರ್ ಆಗಿ ನಡೆಯಲ್ಲ, ಕಣ್ ಕಾಣಲ್ಲ ಸರಿಯಾಗಿ, ಬ್ಲೈಂಡ್ನೆಸ್. ಅಂಡ್ ಶಫಲ್ ವಾಕ್ ಅಂದ್ರೆ ಬ್ಯಾಲೆನ್ಸ್ಡ್ ವಾಕ್ ಇರಲ್ಲ, ಸ್ವಲ್ಪ ಇಂಬ್ಯಾಲೆನ್ಸ್ಡ್ ಆಗಿ ನಡೀತಾರೆ. So all these effects will be seen in a patient who is suffering from tertiary syphilis. Okay, so these are the three stages in the syphilis disease. The first stage is the most common one. So ee stage alli identify detection ಮಾಡಿಬಿಟ್ರೆ very easily you can cure syphilis. But ಏನಾದರೂ ಸರಿಯಾಗಿ ಟ್ರೀಟ್ಮೆಂಟ್ ಕೊಟ್ಟಿಲ್ಲ, ಇಲ್ಲ ಪೇಷಂಟ್ದು ಸ್ಟೇಟಸ್ ಮೇಲೆ ಹೋಗುತ್ತೆ. So it might go to the secondary stage. So secondary stage in a few years the patient will even go to the tertiary level. So these are the three stages. So I have mentioned one more symptom here, that is congenital syphilis. That is when the organism gets transferred from the mother to the fetus, and when the fetus gets affected the symptoms will not be the same. ಸೊ ಅದಕ್ಕೇನೆ ಸೆಪರೇಟ್ ಆಗಿ ನಾವು ಕಾಂಜನೈಟಲ್ ಸಿಫಿಲಿಸ್ ಅಂತ ಹೇಳ್ತೀವಿ. ಸೊ ಹಿಯರ್ ಇಫ್ ದ fetus is affected, many a times abortion will happen or the baby will be born still. So stillbirth ಅಥವಾ abortion will happen. Some cases ಅಲ್ಲಿ live birth ಆಗುತ್ತೆ, that is the baby will be born alive, but it will have signs or symptoms like keratitis, saddle nose or periostitis. Keratitis basically means inflammation in the cornea. So there will be some eye problems in this child. Or saddle nose, where the nasal region would have been affected, nasal trauma ಆಗಿರುತ್ತೆ. Normal bridge ಏನಿರುತ್ತೋ nasal area ನಲ್ಲಿ, that bridge will be collapsed. And periostitis, that is inflammation in the connective tissue that surrounds the bone. So bone ಏರಿಯಾದಲ್ಲಿ ತುಂಬಾ ಇನ್ಫ್ಲಮೇಷನ್ಸ್ ಆಗಿರುತ್ತೆ. So all these conditions will be seen in the baby which is infected from the mother with the syphilis organism. How to diagnose the treponema infection in human beings? See, primary lesion stage alli direct microscopy ಮಾಡಬಹುದು. Here the sample taken will be taken from the chancre lesion itself. Direct agi swab sample ತಗೊಳ್ಬಹುದು. ಅದನ್ನ you just make a wet film on the surface of the slide and then put a cover slip on top of it. And why we do this? Why not staining anta kelbodu? As I said, this organism is very delicate spiral rod. So ನೀವೇನಾದ್ರೂ smear preparation, air dry, heat fix, staining ಇದೆಲ್ಲ ಮಾಡಕ್ ಹೋದ್ರೆ the organism's morphology will be totally affected. So we can't do anything like this. Just like how you see a fungal specimen, you have to see this specimen also. So just wet fill ಮಾಡಿಬಿಟ್ಟು ನಾರ್ಮಲ್ ಮೈಕ್ರೋಸ್ಕೋಪ್ ಅಲ್ಲಿ ಅಂದ್ರೆ ನಾವು ಯೂಸ್ ಮಾಡ್ತೀವಲ್ಲ ಬ್ರೈಟ್ ಫೀಲ್ಡ್ ಮೈಕ್ರೋಸ್ಕೋಪ್ ಅದರಲ್ಲಿ ನೀವು ನೋಡುವುದಾದ್ರೆ ಸ್ಟೈನಿಂಗ್ ಮಾಡಲೇಬೇಕು. Since we don't do staining here, we cannot use a bright field microscope. We have to use a dark ground microscope. So here when we see the organism, just the wet film under the dark ground microscope, you can identify the cells by their spiral structure and also by their movement. ಎರಡಿಂದನು since it is a wet film, movement irutte, spiral structure affect ಆಗಿರಲ್ಲ. So with the help of this you can identify the organism if it is there in the specimen. For diagnosis, the most common technique that is used is this one, serological test, where we will take the blood from the patient and we go for the identification of antigen or antibodies. In this case, we are going to detect the antibodies that is present in the serum. So serological tests, especially for treponema infection, we call it as standard tests for syphilis, STS. Why? Under this test, we usually use an antigen called cardiolipin antigen in the kit. Nammatra kit ಏನಿದೆಯೋ ಅದರಲ್ಲಿ antigen ಇರುತ್ತೆ. Antigen ಯಾವುದು ಅಂತಂದ್ರೆ cardiolipin antigen. I already told you about cardiolipin antigen. This is present in treponema and this is also present in beef heart. Right? So treponema antigen ತೆಗೆದು ಯೂಸ್ ಮಾಡಕ್ ಆಗಲ್ಲ because as I said, antigen ಬೇಕು ಅಂತಂದ್ರೆ organism grow ಮಾಡಬೇಕು, but organism cannot be grown easily in the laboratory. So an antigen which is similar to the antigen present in the treponema, that is what we are going to take, and that is beef heart antigen, which is called cardiolipin antigen. So when we use cardiolipin antigen, we call it as standard test for syphilis. So under this we have two important tests that have been developed. One is the VDRL test. VDRL stands for Venereal Disease Research Laboratory. ಈ laboratory first ಈ test design ಮಾಡಿರೋದು, that is why it is called like that. And this is basically a flocculation test. ಅಂದ್ರೆ ನಾವು antigen antibody reaction ಆದಾಗ we get different types of results. Based on that, we call them as precipitation or agglutination or flocculation. So precipitation andre antigen antibody react agirutte, but you cannot see it clearly with your naked eyes, whereas agglutination alli antigen antibody reaction agirutte and you can also very clearly see it with your naked eyes. Idar eradakku madhyadalli ಒಂದು reaction barutte, that is called flocculation test. So here you cannot see the reaction or the complex with your naked eye, but you can use a microscope and see the antigen antibody complex. Nodakagutte, and that is why it is called flocculation test. And this is a non-triponimal test. Why is it called non-triponimal test? Here the antigen that is used is not taken from the organism itself. It has been taken from some other cell. As I said, beef heart cell in the tegdirodu, which is similar to triponimal antigen, right? So that is why this test is called non-triponimal test. And here in this test, we are basically detecting the antibodies against triponema that is present in the serum. Antibody serum allirutte. This antibody will react with the treponema antigen. So even detect ಮಾಡೋದ್ರಿಂದ this test is basically an indirect way of identifying the infection in the patient. And for doing this test, we need, of course, we need VDRL shaker and we also need a microscope. ಈ test ಹೇಗೆ ಮಾಡ್ತಾರೆ ಅಂತಂದ ಈ YouTube link ಅಲ್ಲಿ ಇದೆ. Please go through this link to understand how this VDRL test is done. This is a very important test. You should be knowing how it is done. Quite simple. Nodidre tumba easy agi artha agutte. So please go through that video. So let me repeat. Serological test antandre, when we use serum to detect the infection in the patient, we call it a serological test. Under that, we have standard test for syphilis. So standard test for syphilis erade, one is VDRL, the other one is RPR. So ee test alli navu yen use ಮಾಡ್ತೀವಿ ಅಂತಂದ್ರೆ, we are using cardiolipin antigen which has been taken from beef heart. And one such test is VDRL test. This is basically a flocculation test where an antigen which is not actually taken from a treponema cell is used, and here in this test we basically detect the presence of antibodies in the serum of the patient. So another non-treponemal test where we use cardiolipin antigen is RPR test. In fact, ನೀವು ಇವಾಗ ರೀಸೆಂಟ್ ಅಂದ್ರೆ VDRL ಫಸ್ಟ್ ಬಂದ ಟೆಸ್ಟ್, ಅದಾದ ಮೇಲೆ ಬಂದಿರುವ ಟೆಸ್ಟ್ RPR. ಸೊ ನೀವೀಗ ಲ್ಯಾಬೊರೇಟರಿ ಎಲ್ಲಾದ್ರೂ ಹೋದ್ರೆ ಸಿಫಿಲಿಸ್ ಟೆಸ್ಟ್ ಮಾಡಿ ಅಂತ ಹೇಳಿದ್ರೆ ದೇ ಯೂಶುವಲಿ ಡೂ RPR ಟೆಸ್ಟ್. RPR stands for Rapid Plasma Reagin Test. Just like VDRL, here also this is a flocculation test. Just like VDRL, here also we are detecting the antibodies present in the serum. But there are certain differences between VDRL and RPR. What is the difference? See, in the VDRL test, we are using pure cardiolipin antigen. We don't mix anything else. Whereas in this test, we are going to mix the cardiolipin antigen along with carbon particles. So ನೀವು ಕಿಟ್ ಸೊಲ್ಯೂಷನ್ ನೋಡಿದ್ರೇನೆ ಬ್ಲಾಕ್ ಕಲರ್ ಅಲ್ಲಿ ಇರುತ್ತೆ, ಮೇನ್ಲಿ ಬಿಕಾಸ್ ಆಫ್ ಕಾರ್ಬನ್ ಪಾರ್ಟಿಕಲ್ಸ್. ಸೊ ಈ ಕಾರ್ಡಿಯೋಲಿಪಿನ್ ಆಂಟಿಜನ್ ಜೊತೆಗೆ ಕಾರ್ಬನ್ ಪಾರ್ಟಿಕಲ್ಸ್ ನ ಮಿಕ್ಸ್ ಮಾಡಿ ಹಾಕಿರ್ತಾರೆ. ವೈ? ಸೊ ದಟ್ we can see the reaction with our naked eyes. As I said, VDRL test na naked eyes alli nodakagalla, you need a microscope anta helide alva. So here antigen jotege carbon particles mix ಮಾಡಿರೋದ್ರಿಂದ easy ಆಗಿ ನಿಮಗೆ with your naked eyes you can see whether the reaction has taken place or not. So here there is no need of any microscope. Both type of test can be done. Qualitative test ಮಾಡಬಹುದು, semi quantitative test. ಏನದು ಡಿಫರೆನ್ಸ್ ಏನು? ಕ್ವಾಲಿಟೇಟಿವ್ ಟೆಸ್ಟ್ ಅಂತಂದ್ರೆ ಸೀರಮ್ ಅಲ್ಲಿ ಆಂಟಿಬಾಡೀಸ್ ಇದೆಯಾ ಇಲ್ವಾ, ಪ್ರೆಸೆನ್ಸ್ ಆರ್ ಆಬ್ಸೆನ್ಸ್ ಅನ್ನು ನೋಡಕ್ಕಾಗುತ್ತೆ. ಸೆಮಿ ಕ್ವಾಂಟಿಟೇಟಿವ್ ಟೆಸ್ಟ್ ಏನು ಅಂತಂದ್ರೆ ಇಫ್ ಆಂಟಿಬಾಡೀಸ್ ಆರ್ ಪ್ರೆಸೆಂಟ್, ಇನ್ ವಾಟ್ ಕಾನ್ಸಂಟ್ರೇಷನ್ ದೇ ಆರ್ ಪ್ರೆಸೆಂಟ್ ಅದನ್ನು ಫೈಂಡ್ ಔಟ್ ಮಾಡಕ್ಕಾಗುತ್ತೆ. ಸೊ ದಟ್ ಇಸ್ ಕಾಲ್ಡ್ ಸೆಮಿ ಕ್ವಾಂಟಿಟೇಟಿವ್ ಟೆಸ್ಟ್. ಸೊ RPR ಕ್ಯಾನ್ ಬಿ ಡನ್ ಬೋತ್ ಕ್ವಾಲಿಟೇಟಿವ್ಲಿ ಅಂಡ್ ಆಲ್ಸೋ ಸೆಮಿ ಕ್ವಾಂಟಿಟೇಟಿವ್ಲಿ. ಸೊ ಈ ಟೆಸ್ಟ್ ಹೇಗೆ ಮಾಡ್ತಾರೆ ಅಂತ ಈ ವಿಡಿಯೋ ಲಿಂಕ್ ಅಲ್ಲಿ ಇದೆ. ಪ್ಲೀಸ್ ಗೋ ಥ್ರೂ ದಟ್ ಟು ಅಂಡರ್ಸ್ಟ್ಯಾಂಡ್ ದ ಟೆಸ್ಟ್. So let me repeat, basically both RPR and VDRL are non-triponimal tests. That means they are not using triponimal antigen. They are using an antigen taken from some other cell which is similar to the triponimal antigen. One. Second, both are flocculation tests. That means antigen antibody reaction ashtu easy agi namage kaanalla. You have to either use a microscope or you have to mix carbon particles along with the antigen to see the antigen antibody complex. So this is second. Third is both tests are going to detect the presence of antibodies present in the serum of the patient. ಎರಡು ಸೇಮ್ ಅದನ್ನೇ ಮಾಡೋದು. ಸೊ ಕಿಟ್ ಅಲ್ಲಿ ಏನಿರುತ್ತೆ ಅಂತಂದ್ರೆ ಆಂಟಿಜನ್, ಕಾರ್ಡಿಯೋಲಿಪಿನ್ ಆಂಟಿಜನ್ ಇರುತ್ತೆ. ಯೂಶಲಿ ಈ ಆಂಟಿಬಾಡೀಸ್, ಈ ಕಾರ್ಡಿಯೋಲಿಪಿನ್ ಆಂಟಿಜನ್ ಜೊತೆಗೆ ರಿಯಾಕ್ಟ್ ಆಗುತ್ತೆ ಅಲ್ಲ, ಈ ಆಂಟಿಬಾಡೀಸ್ಗೆ ನಾವು ರಿಯಾಜಿನ್ ಆಂಟಿಬಾಡೀಸ್ ಅಂತ ಹೇಳ್ತೀವಿ. ಪ್ಲೀಸ್ ಕೀಪ್ ದಟ್ ಇನ್ ಮೈಂಡ್. ದ ಆಂಟಿಬಾಡೀಸ್ ದಟ್ ರಿಯಾಕ್ಟ್ ವಿತ್ ದಿ ಕಾರ್ಡಿಯೋಲಿಪಿನ್ ಆಂಟಿಜನ್ ಇಸ್ ಕಾಲ್ಡ್ ರಿಯಾಜಿನ್ ಆಂಟಿಬಾಡೀಸ್. So I told you there are two non-triponimal tests, which is VDRL and RPR. Why they are called non-triponimal? Because the antigen that is used there has not been taken from treponema. But since the antigens are not taken from the treponema itself, they are non-specific in nature. Sometimes false positive result baruva chances ಇದೆ. So sometimes confirmation ಗೆ triponimal test ಕೂಡ ಮಾಡ್ತಾರೆ. This is not available that easily. So ಈ ಟೆಸ್ಟ್ ಅಲ್ಲಿ the triponimal antigen itself is used. So costly irutte idu because triponema grow ಮಾಡಿ ಅದರಿಂದ antigen extract ಮಾಡಿ ಅದನ್ನ kit ಅಲ್ಲಿ use ಮಾಡಬೇಕು. So it will be quite costly. So there are many such tests, confirmatory tests ಇದೆ which are specific in nature. Here I have mentioned 3 such tests: TPI test, TPIA test, TPHA test. Treponema pallidum immobilization test, Treponema pallidum immunoadherence test, and Treponema pallidum hemagglutination test. So ಇದಕ್ಕೆ ಟ್ರಿಪೋನಿಮಲ್ ಆಂಟಿಜನ್ ಎಲ್ಲಿಂದ ಸಿಕ್ತು ಮೇಡಮ್, ಗ್ರೋ ಮಾಡಕ್ಕೆ ಆಗ್ತಿಲ್ಲ ಅಂದ್ರಲ್ಲ ಅಂದ್ರೆ, so as I said there are group specific antigen. So ಈ group specific antigen ನ non pathogenic ಟ್ರಿಪೋನಿಮಾದಿಂದ ಎಕ್ಸ್ಟ್ರಾಕ್ಟ್ ಮಾಡಕ್ ಆಗುತ್ತೆ. There are certain strains like Nicole strain anta helide, Reiter strain anta ide. Ee strain anna ಸ್ವಲ್ಪ ಕಷ್ಟಪಟ್ಟು you can grow them in the laboratory using cell lines or using rabbit tests. So ಅಲ್ಲಿಂದ antigen ನ extract ಮಾಡಿ ಈ ಟೆಸ್ಟ್ ಗಳಿಗೆ ಯೂಸ್ ಮಾಡಬೇಕಾಗುತ್ತೆ. So because of all these reasons, these tests are not very common and they are quite costly also. So coming to the treatment of syphilis, if it is early stage of syphilis, andre primary stage alle idre, very simply you can treat the patient. Just one single injection of 2.4 million units of penicillin injection is enough. Intramuscularly if you give, that itself will take care of the infection without any problem. But if treatment is not given on time, or if the patient is weak, if the syphilis goes to the next stage, late syphilis alli andre secondary stage ge enadru hodre, the treatment has to be continued for 3 weeks. Weekly once ಈ injection ಕೊಡ್ತಾ ಇರಬೇಕು, 3 weeks ಗೆ ಕೊಡಬೇಕು. Sometimes some patients will be allergic to penicillin, so ಅವರಿಗೆ penicillin ಕೊಡಕ್ ಆಗಲ್ಲ, ಅಂದ್ರೆ intramuscular injection ಕೊಡಕ್ ಆಗಲ್ಲ. So in such cases they use other antibiotics, macrolide antibiotics use ಮಾಡ್ತಾರೆ. ಅದರಲ್ಲಿ two very common macrolide antibiotics that are used are erythromycin and azithromycin. ಎರಡರಲ್ಲಿ ಯಾವುದೋ ಒಂದು ಕೊಡಬಹುದು. In the place of penicillin ಬದಲು you can either give erythromycin or azithromycin can be given. But keep in mind that in a few cases recent days we are seeing resistance to macrolide antibiotics among Treponema organism. So Treponema ಇಲ್ಲಿವರೆಗೆ penicillin ಗೆ resistance ತೋರಿಸಿಲ್ಲ, but macrolide antibiotics ಗೆ resistance ತೋರಿಸೋಕೆ ಶುರು ಮಾಡಿಯಾಗಿದೆ. So prevention is very simple. When we know how it gets transmitted, preventing the disease is very, very easy. One is, we know that they get transmitted through sexual contact. So you have to avoid sexual contact with an infected patient. Okay, so if you come to know that the patient, your male partner or your female partner is infected, avoid sexual contact with such a person. Second is, use physical barriers like condoms. If at all you have to, if you cannot avoid sexual contact, then you will have to use condoms or some such physical barriers, or even antiseptics can be used before the contact. So this can minimize the risk, and only general measures are there. No vaccines are available till now. We don't have any special measures, or we don't have any vaccines available. But apart from this, through transplacental route also it gets transferred, through blood transfusion also it gets transferred, right? So that is why if you go to blood donation centers or blood banks, as soon as they draw the blood, they will do certain tests. Especially nowadays they do triple test anta ಮಾಡ್ತಾರೆ. One is HIV test, second is syphilis test, and third one is hepatitis B test. ಮೂರು sexually transmit ಆಗುವ infections very commonly. So ಈ triple test ಮಾಡಿ ಬ್ಲಡ್ ಡೋನರ್ಗೆ ಏನು ಇನ್ಫೆಕ್ಷನ್ ಇಲ್ಲ ಅಂತ ಕನ್ಫರ್ಮ್ ಆದ ಮೇಲೆನೆ ಬ್ಲಡ್ ಡ್ರಾ ಮಾಡಿ ಸ್ಟೋರ್ ಮಾಡ್ತಾರೆ. So like this, certain general measures can be taken. In fact, pregnant ladies ಅಲ್ಲಿ transplacental infection avoid ಮಾಡಕ್ಕೆ pregnant ladies ಅಲ್ಲಿ every trimester ಅಂದ್ರೆ first 3 months, next 3 months, then last 3 months. Every trimester they carry out this triple test. ಏನು ಹೇಳಿದೆಯಲ್ಲ, HIV, hepatitis B and also syphilis. ಈ ಮೂರು ಇನ್ಫೆಕ್ಷನ್ ಇದೆಯಾ ಇಲ್ವಾ ಅಂತ ಟೆಸ್ಟ್ ಮಾಡ್ತಾರೆ. ಯಾವುದಿದ್ರು ಅದಕ್ಕೆ ತಕ್ಕ ಟ್ರೀಟ್ಮೆಂಟ್ ಕೊಟ್ಟು ಆಮೇಲೆನೆ ಡೆಲಿವರಿ ಮಾಡಿಸ್ತಾರೆ. So all these measures, general measures, if they are taken properly, then you can easily avoid the infection. So the last one is epidemiology. Syphilis is one of the major sexually transmitted diseases, and it is basically a strict human pathogen. That means it does not naturally occur in animals. ನಾವು ಏನೋ ಲ್ಯಾಬೊರೇಟರಿಯಲ್ಲಿ ರ್ಯಾಬಿಟ್ ಟೆಸ್ಟಿಸ್ ಅಲ್ಲಿ ಗ್ರೋ ಮಾಡಿಸ್ತೀವಿ ಅಷ್ಟೇನೆ ಹೊರತು ನಾರ್ಮಲಿ ಆನಿಮಲ್ಸ್ ಅಲ್ಲಿ ನ್ಯಾಚುರಲ್ ಕಂಡೀಷನ್ ಅಲ್ಲಿ ಇದು ಇರಲ್ಲ. ಓನ್ಲಿ ಹ್ಯೂಮನ್ ಟು ಹ್ಯೂಮನ್ ಟ್ರಾನ್ಸ್ಮಿಷನ್ ಅಷ್ಟೇ ಇರುತ್ತೆ. ಹ್ಯೂಮನ್ ಇಸ್ ದ ಓನ್ಲಿ ಸೋರ್ಸ್ ಫಾರ್ ದಿಸ್ ಪ್ಯಾಥೋಜನ್. And as I said, through sexual contact or through personal contact. Personal contact ಅಂತಂದ್ರೆ ಅವರ ಲೀಷನ್ಸ್ ಏನಾದ್ರೂ ಮುಟ್ಟಿದ್ರೆ ಅದರಿಂದನೂ ಬರುವ ಚಾನ್ಸಸ್ ಇದೆ. ಇನ್ ನ್ಯೂಟ್ರೋ ಅಂದ್ರೆ ಟ್ರಾನ್ಸ್ಪ್ಲಾಸೆಂಟಲ್ ರೂಟ್ ಅಲ್ಲಿ from blood transfusion, from organ transplantation, you might get the infection. So these are some ways of basically you have to come into contact with the blood or other body fluids to get this infection. Okay, and there is an estimated 12 million new infections every year that is happening. Since we have good treatment and since the organism can be easily controlled, we don't have much of a problem nowadays. And antenatal syphilis is as high as 30%. So that is before birth infection agodu almost 30% infection rate is seen. So these are some of the points about syphilis. Please go through the whole video presentation carefully and also go through the YouTube videos to understand the diagnostic procedures, because they are very important. They will be asked in the question papers frequently. So go through all that. If you have any doubts, you can always contact me. Thank you.